So ultimately, all variation of the population was initially caused by mutation. Okay? And it's something worth, you might ask then, well, how frequent are mutations? So there's different types of mutations, and their frequencies differ. The mutation you're probably thinking about, or you might be thinking about, is a change in a single nucleotide. So that is to say, we imagine we have our DNA, and it's A, A, C, G, A, T, T, dot, 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 where the dots represent, this is a very long string of nucleotide characters in your genome. And mutation is an error in replication. So it would be, an example would be, this might be the form of the DNA before replication, then after replication, maybe you had A, A, T, G, A, T, T. This is called a point mutation. It's an error that causes the miscopying of a single nucleotide. And it has a rate at which it normally occurs in humans. So in humans, the rate of this type of error is about 1 in a billion. About 1 in a billion. So that would be an error rate of roughly 10 to the minus 9. That's how you'd actually write it in scientific notation. So the normal point mutation rate in humans is about 10 to the minus 9. Right, so what does that mean? It means every time one of your cells replicates, because your genome's roughly 3 billion nucleotides in length, it means that you introduce with every replication about 3 point mutations. Right? There will be 3 differences on average from one copy to the next. And it doesn't mean that every single time you have a replication that you're going to have exactly 3 errors. You might have 0, you might have 1, you might have 2, you might have more, right? But on average, that's how many mutations you occur. Now, some of these mutations are, it depends on which cells these mutations occur, whether they're interesting to you or interesting to population geneticists, right? So mutations that occur in what are called somatic cells, skin, liver, brain, these are of interest to you because these are the types of mutation. A mutation is ultimately the cause of cancer, right? That's bad, okay? So if the mutations happen to occur in some tissue, then it's definitely a concern to you as an individual. They become interesting to population geneticists when the mutations occur not in the somatic cells, but rather in what's called the germline cells. These are cells that ultimately give rise to egg or sperm. So these are the ones that can be passed down, these mutations that can be passed on to your offspring. Okay? And those are mutations that are of potential evolutionary significance because they're going to be the mutations that are introduced in the next generation of individuals in the population. Hope that distinction's clear. Right, but besides point mutations, there's other types of mutations that can occur. So you can have things such as, maybe I'll kind of make a list of them to make it clear. I mentioned point mutations. You can have deletions. This is when the genetic material is lost. You saw already a couple of examples, or I mentioned a couple of examples of alleles that are formed by deletions. Usually they have, it's not a universal convention, but usually a convention is to start the name of the allele with a delta. Okay? And some of these mutations are quite interesting. For instance, there's what is it called? I think it's called a delta 32 mutation in a particular gene that is a protein that HIV has to latch onto in order to get into a human cell. Right? It turns out that individuals who are homozygous for this delta 32 mutation in this particular gene are much more resistant to getting HIV, that the virus cannot get into or has a very difficult time getting into the cell. Right? So this is an example of mutation that is of significance. It's at higher frequency in Northern Europeans, as an example. Point mutations, deletions, you can have insertions or duplications. This is when new genetic material is either inserted into the middle of a string of nucleotide characters or where entire bits of the genome are copied. So for instance, you can have a duplication of a single gene, or you can have a duplication of an entire chromosome. So you've heard of probably trisomy 21. It's the cause of Down syndrome. Well, these are individuals who have 3 copies of the 21st chromosome. There's a mistake during meiosis that causes this. Okay? And it's not like they have any particular mutations in any of the genes on the 21st chromosome. They just have too much expression of all the genes on the 21st chromosome, by about half. Okay? Or you can have entire genomes become duplicated. Right? This is something it's called polyploidization. It's something that occurs a lot in plants. So you can have bits of the genome duplicated, entire chromosomes repeated or duplicated, or even the entire genome. So these are just giving you an idea of the types of mutations that are out there that ultimately contribute to the genetic variations in a population. Okay, that's all I really want to say about mutation. Don't have time to talk about natural selection. Let's move on to natural selection. We already discussed natural selection when I discussed Darwin and the origin of species, so you already have an idea of what it is. But I'm going to describe it a little bit more. So natural selection is the differential reproduction or survival of individuals. And as population geneticists, we want to concentrate on genotypes. So we'll go back to our usual big A, big A, big A, little a, little a, little a. So these are the genotypes, the differential survival and reproduction of individuals with these different genotypes. Okay? We have to introduce this notation of this idea of fitness. It can be a mathematically explicit concept, that is to say, it's a number that you associate with different genotypes that expresses their differential survival or reproduction. But right, so it's just an idea that some individuals, the genotypes with the highest fitness, are going to leave more offspring in the next generation. So if, for instance, as an example, we can talk about positive selection. And so I'll give you an idea of what positive selection is first. That is where individuals with at least with a particular allele are unambiguously better than the other genotypes. So for instance, an example would be, let's say you're an individual, and if you have the big A allele, you're better. You have a higher fitness. And the more big A you have, the more better you are. All right? So if this is an okay individual, this guy's better, and this person or individuals with this genotype are the best in terms of fitness. They survive best, they leave more offspring. So what happens in a population that's under the influence of positive natural selection? Well, if a population is initially composed all of little a individuals, like I have drawn here, and you have a mutation that changes one of those little a alleles into a big A allele, well, this guy now has a fitness advantage. This individuals with this genotype have a fitness advantage. They'll either survive to reproductive age at a higher frequency with a higher probability, or when they do survive to reproductive age, they'll leave more offspring. Right? So you'll find that over time, the composition of the population will begin to change. You'll find more and more individuals that have the big A allele. Initially, most of those individuals will be in the heterozygous state, because initially the big A allele will be rare, but as time goes on, you'll find more and more of the individuals in the population are big A, big A. Okay? And so forth, until all the individuals, over time this will occur, until all the individuals in the population are big A, big A. Okay? That's what will happen when the fitnesses are this sort, when you have what's called positive natural selection occurring. Now, when the big A allele is finally at a frequency of 1, that is to say all the individuals in the population are homozygous with the big A allele, the mutation, the big A allele is said to be fixed. Okay, so this process, you start off with a mutation little a, big A increases the frequency until finally its frequency is 1. At that point, the big A allele is said to be fixed in the population. Okay. Where am I going here? And I should also mention, or repeat, I suppose, that these fitnesses, once again, are context dependent. You change the environment. It could be that a genotype, maybe big A, that was best in one environment, maybe it's not best in another. So, for instance, the delta 32 mutation is only best in the presence of HIV. The S allele is only the best in the presence, or is only, even natural selection only cares about it in the presence of malaria. Okay? So it is all context dependent. So a good example of directional selection in human populations is lactose tolerance. Okay? So what do I mean by lactose tolerance? This is the ability to digest lactose as adults. So I'm one of those individuals that can do that. It turns out that's fairly rare in mammals. Okay, so in mammals, obviously, young or fed milk, that's one of the characters that diagnoses mammals. But eventually, once the offspring are weaned, the particular gene is shut off as an adult. So it's only active in the young, it's turned off in the adult. So let's talk a little bit about what the gene is. So there's the gene that's involved is called lactase fluorescein hydrolase, or LPH. I have the, I don't think actually, I do have it written in my notes, so I'm not going to write it again, but LPH is the gene that's responsible. Once again, normally this gene is active in young when they're feeding on the mother's breast milk, but eventually as adults this gene is turned off. Okay? There are a couple of cases in mammals where this gene is constitutively active in adults. One example is cats. House cats can drink milk and digest it. And the other is some populations of humans. Okay? So how many people here in this room can digest milk right now? Yeah, so a lot of people are raising their hands. So it's not clear whether you really can or can't. So there's actual tests, I'm not going to describe them because they're kind of gross tests, that you can perform on a person to see if they really can, if they really are lactose tolerant. All right? And it turns out there's really only a couple populations of humans that can do this. Northern Europeans, not all, but many, and populations in Eastern Africa. There's individuals in populations in Eastern Africa that can also digest milk as adults. Okay? And so a lot of you that think you can probably really can't, or maybe it's just starting, you're going to be noticing it over the next few years. You'll notice it because whenever you have ice cream or a lot of milk, you'll become excessively flatulent. So you'll know it when it occurs. Screen blank. So we're having a few technical difficulties. Oh, all right, the technical difficulties weren't there, it was with me. All right, so we're back here. It turns out you actually have to have the computer plugged into the cord here. Okay. So this is a study that came out about 6 years ago now. It's people are trying to discover what mutations were responsible for lactose tolerance in humans. And so what you see here is a figure directly from that paper. And from going from the top to the bottom, you're getting more and more focus. So on the very top, you have laid out a 3 megabase. There's a 3 million nucleotide segment of the second chromosome is laid out on the very top. And you can see that there's lots of little blue dashes. Those blue dashes represent nucleotides in the genome that are known to vary from one individual to another in the human population. These are those SNPs that I talked about, SNPs, right? Single nucleotide polymorphisms. But as you go, you can see that they've focused in on one part of that 3 megabase section. You can see that what they've labeled there is the different genes that you can find in that region. Then they focused in on just this region, with the dashes being even more fine-scale variation that people are aware of. And finally down to parts of the non-coding region in this LPH gene. And you can see these are the single nucleotide polymorphisms that have been marked. You have G and C, that's one of the SNPs. Some individuals have the G nucleotide at that position, that's the wild type or the ancestral condition. And then you have C being the derived or the mutant form. Here's another SNP, another SNP, another SNP, and then another one that's just in place there. And then here, just to remind me so I don't screw up, these SNPs, the ones that I marked with A, are the ones that are responsible for making this gene stuck on, essentially, in Africans. And there's a different mutation at this position, 13,910, that's the one that's responsible for keeping this gene stuck on in Europeans. So the first point I want to make here is that this is an example of what's called convergent evolution, the independent evolution of a particular trait. It turns out that both of these societies have what's called a pastoral society. They raise cattle, they drink lots of milk and yogurt. Okay, think about Swedes and how much yogurt those guys eat, right? Well, in those societies, being able to digest milk as an adult is a real selective advantage. It's a huge source of calories for many populations. So it appears that natural selection is responsible for driving these different alleles to high frequencies in these two different populations, and it's happened independently. Okay, if it were the same genes, it was the same mutations that was responsible for lactose tolerance in these two populations, Africans and Europeans, then it would indicate one of two things. Either there's a very limited number of ways you can stick this gene on, right? There's only one mutation or a handful, and they just both happen to independently find the same mutation, or you had a common ancestor. That is to say, there's the common ancestor of the individuals that have this had this particular gene stuck on. Those are the two different possible explanations. But because we don't see that, it's very clear signature of the independent origin for lactose persistence, or lactose tolerance in these two different populations. Okay, so that's a good example, I think, kind of a fairly recent one of natural selection in humans. So we're doing on time. All right, I think I can go just a little bit more. I want to talk about kind of the flip side of positive selection. Positive selection is called purifying selection. So over here, when I talked about LPH or positive selection, having one of the copies, as an example, the big A allele was unambiguously good. The more of that big A allele you had, the higher your fitness, right? Well, the opposite is called purifying selection. And this is the case for most mutations. When they occur, they're bad, right? So it's only a small fraction we think of all mutations that confer a selective advantage, that is to say that they're beneficial. Most mutations when they occur are deleterious, right? Think about it this way. You take a perfectly functioning protein and you randomly change it. What do you think your chances are of improving that protein? They're small, right? So most mutations when they occur, you're breaking something. You're not fixing it, making it better, you're breaking something. So as an example, if you have a mutation that causes this big allele to become a little allele, the chances are that this individual isn't going to have a higher fitness, although that could occur occasionally. But it's usually the case that you've broken something that was working just fine, and this individual has a lower fitness. What does natural selection do in these cases? It tends to remove those individuals from the population that have lower fitness. They leave less offspring in the next generation, or they survive with a lower probability to reproductive age. So natural selection in this case removes deleterious mutations. So natural selection removes deleterious allele. Right, so you would think that would be the end of the story. So why do we have any of these rare genetic diseases at all? Well, the problem is that mutation is constantly introducing these deleterious mutations to a population. They say, well, geez, mutation is really rare. Yes, it is, but you also have to think about there being a large number of genes in a particular genome. There's 30,000 targets for mutation in a single individual. And not only that, populations can be large. So every generation in a population, you're going to have new deleterious mutations being introduced. Okay? So you have mutation, putting them in, natural selection, removing them. The frequency at which you see a deleterious mutation in a population is often under what's called, or at what's called mutation selection balance. All right, so there's a balance struck between the rate at which these mutations are introduced and the rate at which natural selection removes them. Mildly deleterious mutations can be at a higher frequency because natural selection has a harder time scrutinizing mildly deleterious mutations. If the mutation results in a highly deleterious allele, that is to say you're a very, very sick individual or you die, well, the natural selection can more finely scrutinize those types of mutation and more efficiently remove them from the population so the overall frequency of that allele is smaller. Okay? Anyway, so I'm going to break here, and we're still a little bit behind my normal schedule, but we'll catch up next week, next lecture.